Hello, biology. Happy Thursday. Welcome back. It's Thursday, yeah. It's Wednesday tonight. I'm recording this. Uh, miss you guys. Getting better, but I'm still not quite good enough to come to school all day. This is the last part of Chapter 9. It's going to be more Punnett Squares. We're going to talk about some special kinds of Punnett Squares. And for my couple of you that are in forensics, you guys will be able to help out the other biology kids with these because you guys have already done some of them. So the first special instance we're going to talk about is incomplete dominance. Mendel's peas were really nice because they were really simple genetics. They didn't have any trickery to them. Unfortunately, genetics is not always that nice and dry and clean. Mendelian genetics are what we call these simple Punnett squares. And in humans, they're a lot, your genetics are a lot trickier. Like there's 6 or 7 genes that code just for eye color, let alone your hair, your melanin type, all the different things that make you you. There's 14 genes in height. There's a lot of stuff going on in the human body. But these ones we've been looking at is complete dominance, where if we have, let's say we have a purple plant and a white plant. The purple plant, the purple gene is completely dominant. That means that both PP, big P, little p, and big P have the same color, which will be purple, and then little p would be white. Now, in incomplete dominance, does not always happen. This is when the phenotype of the heterozygote, so the one that has a big letter, little letter, is intermediate between the dominant and recessive. And intermediate means in the middle. So let's use, for example, a red flower and a white flower. So big R is red, dominant red. Little r is recessive white. Now, in incomplete dominance, this is where it gets tricky. Big R, little r would be pink. So let's do an example. Let's do, you know what? For the sake of just getting some cool things out there, let's do a heterozygous. Let's do a pink flower. So pink flower is big R, little r. And let's mix it with a white flower, little r, So like before, we're going to pull this big R down, this big R down, this little r down, and this little r down. And we're going to put this little r across, and this little r across, and this little r across, and this little r across. Now we have 0 red for our genotype here. Genotype ratios. We have 0 red. We have 2 of the heterozygous, 2 of these pinks, and we have 2 whites. 2 of these whites, these little r's. So in between these 2 flowers, we had a pink flower and a white flower. Half of them will be pink and half of them will be white. Now, codominance is a little bit different. In codominance, both brown and black have capital letters. Both of these are dominant. So this is how we get calico cats. If you've ever seen a calico cat, they're really pretty. They have 3 colors on them. They're always female. When I come back, we'll talk about sex-linked traits. Color blindness in humans is sex-linked. There's lots of things that are sex-linked that ride on the X chromosome. All right, so in calico cats, let's say, for example, I have a brown cat that's a heterozygote, big B, little b. This is the brown color, this is the white, and then I have a black one that's a heterozygote, which is big L, little b. Now, this BL here, this is the calico. This is the one that's going to have both brown and black, and then where that spots it doesn't is going to be that white that shines through when these guys are overlapping. So this ends up having both the brown and black instead of not just having one or the other. So this is how we get calico cats. This one with the big B, little b is going to be just a brown guy. This guy with the big L, little b would be black. And these two could have a white baby as well. So even though they both have a dominant trait, because they have that recessive gene hiding underneath, you can have a white recessive baby as well. Okay, so in humans, we have codominance. with our blood types. Both A and B blood types are capital, and then O is the little one. This is why we can have blood types like AB. In this first cross, however, we're gonna keep it simple. We're gonna start slow. We're gonna have an O blood type, and we're gonna mix with somebody who we know has blood type AO. Big A, little O. So bringing this A down, bringing this O down, bringing this O across, and bringing this O across. The probability of blood type A, 50%. 2 out of 4. Blood type O, 50%. 2 out of 4. Everything else, big ol' zeros. 0 for B and 0 for AB. We don't have either of these types. Okay, I'm kind of in the way here, but that's okay. I think I can function. I'll move myself up to the top right so you'll see me on the top right. So typing A with O. So we don't know, is it type AA? This is one of those test cross that we did before. Or is it type AO? So if we know what the offspring are, we can figure out what mom and dad were. So in this case, this first one, big A, big A, big A, big A, and then OO, OO. So we have 100%, 4 out of 4 of these offspring are A. So this gives us a pretty good idea that whatever parent this is has that AA, that double A. Now on this one over here, you're going to see a much different trend. We have big A, little O, little O. We bring this little O across, and this little O across. And now all of a sudden, out of nowhere, we have 2 out of 4A, not out of nowhere, you guys know better than that, and 2 out of, I'm doing this blind, 4O. So we have a 50-50 split on this one, letting us know that this parent is AO instead of AA. All right, now we have cool thing here, and I should have capitalized this B. Pretend, students, look perfect, capital B. Got it right the first time. So we have AO and BO, not the body odor, the blood type. Crossing these, we're gonna get A, little O, little O, and I'm gonna fudge here a little bit. Big B, getting moved across. Little O, getting moved across. And now we have all 4 blood types. We have 1 out of 4A here. 1 out of 4B here. Because remember, B is dominant over O. We have 1 out of 4O down here. And we have 1 out of 4AB, which AB is the most rare blood type. Here, and AB is codominant, so we see both of those. What blood actually looks like, dear students, is on a blood cell, which look kind of round and like this. On A ones, they have a certain protein. Let's say the A protein looks like this. The B ones have a very different looking protein, like it looks like this. The O ones have nothing on them. Mr. Greer is O negative, which means he has no proteins on the outside of his blood. And then if they are AB, it means that they have both A and B proteins. So their blood has a lot of proteins on the outside of it, where Mr. Greer's is very bland in comparison. All right, for this last example, we have type AB. With type O, when doing these problems, always figure out what blood type you're starting with and what the other one is. So bringing this A down, AA, BB, bringing these O's across, O, 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 O. And then our probabilities, 2 out of 4, 50% A. And 2 out of 4, 50% B. You will have worksheets today on both incomplete dominance and codominance. And then you also have a study guide. Now, I'm hoping to be back tomorrow, Friday, to help administer your quiz. But if not, I believe in you guys. You guys are going to do awesome. I miss you guys. I miss being there. I'm getting tired of rattling around this place all alone. But hope you guys have a great rest of your day. And that you feel pretty confident on these Punnett Squares. All right. Ciao, ciao.